Welcome. Myself, Dr.M.Manigandan, Assistant Professor, Department of Microbiology, Sacred Heart College, Tirupattur. In this video, I would like to explain about the polio virus, its structure, transmission, replication, pathogenicity, clinical syndromes, and treatment and prevention. Historically, the poliovirus infections have been observed from the 18th century. The polioviruses belongs to the largest enterovirus family. The polioviruses are pathogenic to human and animals. The first epidemic outbreak was observed in the year of 1868 at Oslo, Norway, with 14 cases of polioviruses. After that, a number of epidemic outbreaks have been identified. The infectious particle poliovirus was discovered in 1908 by Carl Landsteiner. To prevent the spread of disease, the first poliovirus vaccine was developed by Jonas Salk in 1953. He developed a killed viral vaccine. Later, in 1961, Albert Sabin developed a live attenuated polio vaccine. Structurally, the polioviruses are smaller in nature, as the name itself indicates, pico meaning with ribonucleic acid as its genetic material. The virus is a non-enveloped virus with a naked capsid. The size of the virus is 30 nanometer in diameter with icosahedral capsid. The genetic material present in the virus is a positive sense single-stranded RNA with 7,200 to 8,450 bases. The icosahedral capsid has 12 pentameric vertices, each of which is composed of 5 protomeric units of protein. The protomers are made of 4 virion polypeptides VP1, 2, 3 and 4. The viral proteins 1, 2 and 3 are found in the outer surface of capsid, and the viral protein 4 is found in the inner regions of capsid. Transmission. The virions are transmitted by the fecal oral route. Contaminated food and water are the major source of virion. Contrary to the name, the enteroviruses, they do not usually cause enteric diseases, but they do replicate in intestine. The upper respiratory tract, the oropharynx and the intestinal tract are the portals of entry for enteroviruses. Due to the naked capsid structure, polioviruses are resistant to stomach acid, proteases and bile. Viral replication is initiated in the upper mucosa and lymphoid tissues of the tonsils and pharynx, and the virus later infects the M cells and lymphocytes of the Peyer's patches and enterocytes in the intestinal mucosa. Replication of polioviruses. The polioviruses binds to CD155 receptor molecules of the human host cells with the help of viral protein 1. The poliovirus receptor CD155 is present in many different human cells, but all these cells will not replicate the polioviruses. On binding to the receptor, the viral protein 4 is released and the virion becomes weakened. The genome is then directly injected across the plasma membrane through a channel created by the viral protein 1. As the viral RNA itself resembles mRNA, it directly binds to the cell's ribosomes. The ribosomes recognizes the viral RNA as cellular mRNA. Then it synthesizes a polyprotein containing all viral protein sequences within 10 to 15 minutes of infection. This polyprotein is cleaved by viral proteases encoded in it. The viral RNA-dependent RNA polymerases generates a negative sense RNA template from which the new mRNA genome can be synthesized. The viral proteases helps in cleaving and assembly of structural proteins. Nearly 1 lakh virions per cell may be produced and released through the cell lysis process. The time taken for the entire replication cycle is around 3 to 4 hours. Pathogenesis. The source of virion is contaminated food and water. After the initial replication in the upper respiratory tract through primary viremia, virus spread to reticuloendothelial cells of the lymph nodes, spleen and liver, from where secondary viremia initiated and the symptoms are observed. Most enteroviruses are cytolytic, replicating rapidly and causing direct damage to the target cells. In the case of poliovirus, the virus gains access to the brain by infecting skeletal muscle and traveling up the innervating nerves to the brain. The virus is cytolytic for the motor neurons of the anterior horn and brain stem. The location and number of nerve cells destroyed by the virus govern the extent of paralysis. Clinical syndromes. The incubation period for enteroviruses varies from 1 to 35 days depending on the virus, the target tissue, and the person's age. Viruses that affect oral and respiratory sites have the shortest incubation period. Polioviruses may cause one of the following 4 outcomes. First one is asymptomatic illness. This results if the virus infection is limited to the oropharynx and the gut. At least 90% of the poliovirus infections are asymptomatic. Second one is the abortive poliomyelitis, the minor illness. It is a non-specific febrile illness occurring in approximately 5% of the infected people. Fever, headache, malaise, sore throat and vomiting occur in such persons within 3 to 4 days of exposure. Non-paralytic poliomyelitis is the third outcome. This occurs in 1 to 2% of the poliovirus infected person. In this disease, the virus progresses into central nervous system and the meninges, it causing back pain and muscle spasms in addition to minor illness. The fourth outcome is the paralytic polio. This is a major illness, causes in 0.1 to 2% of the infected persons. This is the most severe outcome. It appears in 3 to 4 days after minor illness subsides. In this disease, virus spreads from the blood to the anterior horn cells of the spinal cord and to the motor cortex. The spinal paralysis may involve one or more limbs, whereas the bulbar or cranial paralysis may involve a combination of cranial nerves and even the medullary respiratory center. Paralytic poliomyelitis. It is characterized by an asymptomatic flaccid paralysis with no sensory loss. The degree of paralysis varies in that it may involve only a five muscle group, example one leg, or there may be a complete flaccid paralysis of all four extremities. The recovery may occur within 6 months, but as long as 2 years may be required for complete remission. Bulbar poliomyelitis. This can be more severe, may involve the muscles of the pharynx, vocal cords and respiration. This may result in death in 75% of the patients. Post-polio syndrome. It is a sequel of poliomyelitis that may occur much later in life, 30 to 40 years later, in 20 to 40% of the original victims. Affected persons suffer a deterioration of the originally affected muscles. Poliovirus is not present, but the syndrome is believed to result from a loss of neurons in the initially infected nerves. Laboratory diagnosis. The clinical samples to be collected are cerebrospinal fluid, feces, and throat swab. The virus grows well in the monkey kidney tissue culture. The exact type of enteroviruses can be determined through the use of specific antibody and antigen assays. Example, neutralization test, immunofluorescence test, enzyme-linked immunosorbent assay, or reverse transcriptase polymerase chain reaction helps in the detection of viral RNA. Detection of specific immunoglobulin M is very much important for confirming diagnosis of polioviruses. Treatment and prevention of poliovirus infections. A new antiviral drug, pleconaril, is available. The drug inhibits the penetration of picornaviruses into the cell by inhibiting the viral protein 1 activity. It is administered early in the course of infection. The prevention of paralytic poliomyelitis is one of the triumphs of modern medicine. This was achieved with the help of two types of poliovirus vaccines. First one is the inactivated polio vaccine IPV developed by Jonas Salk, and the second one is a live attenuated oral polio vaccine developed by Albert Sabin. Both vaccines incorporate the three strains of polio which are stable, relatively inexpensive, and induces a protective antibody response. Thank you for your kind listening and post your comments. Thank you.